Okay, so picture this. You're a genetic detective. You've got this single, mysterious strand of DNA, and your job, your mission, is to find its relatives. Not in a small town, but in a library that contains the genetic code of basically all known life on Earth. So where do you even start? Well, today, we're cracking the case of sequence alignment. These are the powerful tools that let us decode the very language of biology.
So here's our game plan. First, we'll tackle the main challenge, what I call the genetic search problem. Then, we'll figure out how we even score a potential match. After that, we dive into the high-speed tools like BLAST, learn how to make sense of the results, and then zoom out to see how to align a whole family of sequences. And we'll wrap it all up with some pro tips right out of the detective's handbook.
The central challenge here is just immense. You have your query sequence, that's your needle, and you need to find something similar in a database that isn't just a haystack. It's more like a genetic haystack the size of a continent. It's a huge problem. And any search method you use is kind of like a tightrope walk between these three things. You need sensitivity, which means finding all the real relatives, even the super distant ones. You also need specificity to ignore the millions of random sequences that just look similar by pure chance. And of course, you need speed, right? Because nobody has time to wait months for an answer. But here's the catch. You can't have it all. You can't max out all three at once. Every search is a strategic trade-off. This all leads to two main ways of thinking. On one side, you've got your rigorous methods. This is like a detective who literally interviews every single person in the city. It's incredibly thorough, and yeah, it will find the absolute perfect answer, but it's going to take forever. On the other side, you have heuristic methods like BLAST. This is the clever detective, the one who uses shortcuts and rules of thumb to solve the case in a tiny fraction of the time, getting an answer that is almost always just as good.
So, before our clever detective can even start searching, they need a system to rank their leads. What actually makes a good match, especially when the sequences aren't a perfect one-for-one copy. This is where we absolutely need a scoring system.
The first way to score things is called PAM. The best way to think about this is as the theory-driven historical method. Scientists looked at proteins that were super closely related and built a model of how amino acids tend to change, or mutate, over really long stretches of evolutionary time. It's kind of like a historian using patterns from the last century to predict what might happen in the next one.
Now here is a super important point, and honestly it's a bit backward from what you'd expect. With PAM, the higher the number, the more distant the relationship you're searching for. So a PAM 250 matrix is specifically designed to find relatives that split off from each other a long time ago. You'd use that if you think you're looking for an ancient connection between, say, a human protein and a fish protein.
And then we have the second approach, BLOSUM. If PAM is our historian, BLOSUM is the field biologist, out there with boots on the ground. It doesn't start with a big theory of evolution. Instead, it just looks at tons of different proteins and directly observes which amino acid substitutions actually happen in the most important conserved parts of those proteins, these blocks. It's totally data-driven.
And just to keep us on our toes, the numbering for BLOSUM is the complete opposite of PAM. Yep, with BLOSUM, a lower number, like BLOSUM 45, is what you use to find more distant relatives. It's because that matrix was built by looking at proteins that were only about 45% similar, so it's tuned for finding those faint ancient connections. This table really sums it all up. Just look at the basis row. PAM is built on an evolutionary theory, while BLOSUM is pure observation. And check out the data source. PAM uses global alignments, trying to match sequences from start to finish. But BLOSUM focuses on local alignments, those critical conserved blocks. And that local focus is a huge deal. It's why BLOSUM is often the go-to for finding those specific functional hotspots in a protein, which is what researchers are very often looking for.
Okay, so we've got our scoring system figured out. Now let's get to the really cool part, how tools like BLAST actually use these scores to power a high-speed chase through those gigantic databases.
The absolute genius of BLAST is its speed, and it gets that speed by working smart, not hard. Here's how it does it. First, seeding. It chops your sequence up into these tiny three-letter words. Then, step 2, searching. It just zips through the entire database looking for exact matches to those little words. It's only after it finds a promising spot that it moves to step 3 and scores them with something like BLOSUM 62. And only then does it do the really heavy work in step 4, extending the alignment outwards from that starting point. It's like searching a massive book for one specific phrase and then just reading the sentences around it. It's so clever.
And there isn't just one BLAST program. There are different flavors for whatever kind of evidence you're working with. Starting with DNA and looking for other DNA, that's BLAST-N. Got a protein and you want to find its cousins? Use BLAST-P. And then there are the really cool ones, like BLAST-X, which can take your raw DNA, translate it into every possible protein it could make, and then search with all of those. It's like the ultimate multi-tool for a genetic detective.
Okay, so the high-speed chase is done. We have a list of potential suspects. But how good is the evidence really? How do we know if a match we found is a genuine biological relative, or if it's just a random, meaningless coincidence?
The single most important piece of evidence is the E-value. That stands for expectation value. All this number tells you is how many matches with a similar score you'd expect to find just by pure random chance. So here's the bottom line. The lower the E-value, the better. A lower number means your match is more statistically significant. It's basically our detective's confidence score.
So what does a really, really good E-value look like? Well, it looks something like this. What that number means is less than 1 with 50 zeros after the decimal point. It is an unbelievably tiny number. The odds of that happening by chance are practically 0. This isn't just a clue anymore. This is your smoking gun. And here's a handy little decoder ring for when you get your results back. If your E-value is down in that green zone, less than 1 E50, you have found a direct relative. It's a slam dunk. If you land somewhere in the middle, you might have a clue, a possible lead for a very distant relationship. But if you're up in the red zone with an E-value over 10, you're almost certainly looking at random noise. Case closed. Move on.
So far, we've really just been talking about comparing one sequence to another. But the real power, the real insight, comes when you line up an entire family of related sequences all at once. This is called multiple sequence alignment, and it lets us see the whole bigger picture.
So why is this so powerful? Well, think of it this way. A simple alignment is like a conversation between two people. A multiple sequence alignment, or MSA, is like listening in on an entire family reunion. All of a sudden, you can see the shared traits, the family resemblances. You can spot the exact amino acids that are so important, so critical, that they haven't changed in millions of years of evolution. This is how we find the true heart of a protein family.
Now, the classic tool for this is called CLUSTALW. It's brilliant, but it has this one little flaw. It's greedy. It builds the alignment step by step. And if it makes one mistake early on, say it pairs up two distant cousins incorrectly, that error gets locked in and can never be fixed. It's just like putting a puzzle piece in the wrong spot right at the beginning. The whole final picture is going to be off. Once an error, always an error.
So, naturally, scientists figured out a way around this. Newer tools like T-Coffee were developed to solve this exact problem. Before it even commits to an alignment, T-Coffee basically does a ton of homework. It builds this library of evidence, comparing all the sequences in multiple ways. It just puts in more work up front to make a much better first move, avoiding those early mistakes that trap the older algorithms.
All right, we've covered the theory, the tools, the analysis, the whole shebang. To wrap things up, let's go over a few pro tips straight out of the detective's handbook for running a much better investigation.
Okay, rule number 1. If you can, always search with proteins. The 20-letter amino acid alphabet gives you a much stronger signal than the 4-letter DNA alphabet. Second, mask out those low-complexity regions. Think of these as weird repetitive stutters in the sequence that can throw off the search. Third, pick the right scoring matrix, like we talked about. BLOSUM 62 is a great default, but you might need something else. And finally, the golden rule. Never, ever blindly trust the default settings. Understand your case and tune your tools to match it.
You know, when you step back and think about it, these tools are incredible. We can go from finding a single gene in a haystack the size of a continent to reconstructing the entire evolutionary history of a protein family. It's a detective story that's literally written in the language of our own cells. And the only real question left is, with these tools getting smarter and faster every single day, what amazing mysteries are we going to solve next?